So welcome to this lesson, guys. Toda kutarisa nezve plant nutrition. So we are still continuing our series biology. So when we look at plant nutrition, we're going to look at primarily the aspects of food that has to do with the plant, right? So we are dealing with the aspect yekuti a plant redu, it has to be eating. In other words, it has to be having food that it takes in, right? So the plant, unlike people, unlike other animals, we find that the plant redu haritese haridyi chikafu in the normal sense that we eat chikafu. Isusu tinoti kana tikaita nzara tinoenda somewhere else and then we tend to eat that food. So the plant is always sticking on one place, rakabatwa nemidzi pa one place, but at that place pariri papo ndo parinokwanisa kuwana it's what? It's food. And also the other thing is that a plant itself, it is able to produce food. Unlike us, we are unable to produce food, but the plant itself, it has an ability to produce food on its own. So we are going to look at the first aspect, which is known as photosynthesis theory. So that's the basic. So on photosynthesis theory, we are going to touch two aspects on the word photosynthesis. Photo, it means light, and synthesis, it means make up. So in other words, we are saying that this is a process in which we use light to make up food on its own, right? So on our first point, green plants make carbohydrates, the food carbohydrates in form of glucose, from the raw materials carbon dioxide and water. So the two raw materials that the plant uses to make food is number one, carbon dioxide, and number two, it makes it using water. So water and carbon dioxide are two raw materials. So in the absence of water, in the absence of carbon dioxide, we find that our plant is unable to make food. Why is it unable to make food? Because those two are the primary raw materials, right? So on point number two, at the same time, oxygen is made up and released as waste product. So parinogadzira food yaro richishandisa carbon dioxide and water, richabva raburitsawo oxygen as a waste product. So the reaction requires energy, which is obtained by the pigment chlorophyll trapping light from the sun. So what is happening? We are saying that this process photosynthesis kuti we require some sort of energy such that that reaction can occur. So for that reaction to occur, that energy is absorbed by a pigment which is called chlorophyll. So this pigment is a green pigment which gives the leaves its green color. So every leaf that you see that is green, it has what? It has chlorophyll, and that chlorophyll gives the green coloring. That green coloring pigment is the one that traps sunlight energy or that absorbs sunlight energy. So the function for chlorophyll is to absorb sunlight energy. And so photosynthesis can be defined now as the process by which plants manufacture food in form of carbohydrates from the raw materials. So we have to also mention the raw materials. From the raw materials edu, they are what? Carbon dioxide and water. Water, it's H2O, and water using energy from light. So in other words, Trukuti, photosynthesis is defined as the process by which plants manufacture food in form of carbohydrates from raw materials, in brackets, carbon dioxide and water, closing bracket, using energy from the light. So that's the proper definition for photosynthesis. ndo inenge iri definition inenge yakabata zvese. We have included all, and it. So now we can then sum it up to form an equation. An equation shows us the processes, the chemical processes that are there. So the chemical processes that are there, we are saying that carbon dioxide here iri kusangana newater, then pane arrow iri, right, reblue iri. So arrow kana kuri kuma equations ndizvo pakanyorwa paronzi paarrow pobva nyorwa pamusoro nepazasi, it means that those two things are conditions that are necessary for the reaction to occur, right? So which means that we require a condition of sunlight and a condition of chlorophyll, such that water yedu ne carbon dioxide, they can combine together to form glucose. So it forms glucose plus oxygen. So we are saying that these ones are true, ndo are true kuti raw materials, and these ones are what we call the what conditions necessary, and then these ones are the products. So that's what happens. That's the equation for photosynthesis. It gives us the raw materials, the conditions, as well as the products. Let's look at it in depth. So we are saying that carbon dioxide diffuses into the leaf through the stomata. So the stomata are the ones, my openings are kuita kuti carbon dioxide ipinde into the leaf, right? So when carbon dioxide is entered into the leaf, the water is taken up by the roots and transported through the xylem to the leaves. And so tiri carbon dioxide yedu kwairi kubva chi via diffusion from the stomata, and then water yedu iri kubva from the roots, and it is transported through the xylem until it reaches the leaves. Then zvasangana izvozvo zvosangana pane light ne chlorophyll, and then we have carbohydrates. Now these carbohydrates are used to make what substances that the plant needs, right? So they can be formed ari iwo carbohydrates, but after carbohydrates they have to be what? They have to be other substances that the plant would require. And also the other thing is that this glucose is also used by the plant for respiration. So we have two things that this glucose here forms rhinoita. Two main things. It is used by the plant to make other substances that are not carbohydrates. It can be used probably to make proteins, right? So you can find a plant redu rinenge riching provide proteins. Why? Because proteins iwo akagadzirwa from glucose, right? Then we can also have respiration, whereby to plant redu it needs energy for its processes to occur, especially respiration. And it so respiration inoshandisa chi glucose yoyi, because respiration, as we said earlier on, it is the breakdown of glucose to produce energy. Then oxygen yedu, it diffuses out. Oxygen here produced, so it diffuses out through the leaf, out of the leaf through the stomata, and is used by respiration. So it is used for respiration by other organisms, right? So all the other organisms, such as the other animals, they require that oxygen such that they can work out respiration. Then to end up the photosynthesis theory, now we want to look into depth. Takuti photosynthesis here now, it can be written, the process can be written in form of an equation, right? So kana tichinyora in form of an equation, we are saying carbon dioxide yedu yakunyorwa as an equation plus water yedu, then it gives us carbohydrates. So our carbohydrates, the common formula for carbohydrates, that's C6H12O6, then plus oxygen. Then after you have written that, you then have to balance your oxygens, and then you have to put your 6 there. So after you write your equation, you then have to balance it. Then the light energy is converted into chemical energy in bonds that are holding the atoms of glucose together. So we are saying that parikuitika this photosynthesis yedu ndizvo this light energy yedu is being converted into what? Into chemical energy, right? So it's being converted to chemical energy, but this chemical energy yedu yakuwanikwa glucose, it's primarily found within the bonds that hold the molecules together. Is that okay? So when those bonds would break, you then find energy being released. Are we together? So we are saying that the energy is trapped as light, and then as it is trapped as light, it is converted into chemical energy, which is the what? Which is the food that is formed, or the carbohydrates. So common question inowanzobvunzwa kunzi panoitika photosynthesis, what energy change do we see? So the energy change that we see is that it changes from light to chemical energy. Then we go next, product of photosynthesis. So plants use the glucose to make and they make as a source of energy for respiration. So that's number one. They would make use of it as for respiration, kuti ikwanise kuita ma respiratory processes ainenge ichida kuita. Then number two, they can also convert starch. They can also convert the glucose into starch such that it can be stored. Saka kana chida store glucose yedu, it is converted to starch. Why? Glucose yedu no dissolve, starch yedu it doesn't dissolve. So starch yedu it will be more better, and so we are saying that this glucose, let's just write like this, glucose yedu icha convert to starch, and then it is what? Stored. Or this glucose yedu, again, it can be converted into lipids. So my lipids yedu they are more like fats, right? So it can be converted into lipids for an energy source in seeds. So lipids for energy source in seeds. And then glucose yedu iyo, again, it can be converted into cellulose. So as we all know, cellulose is primarily used as a cell wall, right? So cellulose for cell walls. Or it can also be produced into amino acids. Amino acids, and these amino acids are used to make proteins. So as I've said earlier on, so the plant redu rukuda starch, rukuda ma lipids for in terms of my seeds arrow, rukuda futi cellulose in terms of my cell walls arrow, and rukuda futi ma amino acids in terms of the proteins that it would produce. So when combined with nitrogen and other minerals from the roots, ndo painenge chitipa machi ma proteins, and it. So ukatasa the way or the fate of the end product of photosynthesis. So we say kuti is the fate of what? Of glucose, and so to the fate of glucose, where is glucose going to go? And it ga inocheta starch, icheta ma lipids, icheta cellulose, icheta futi amino acid for proteins, and it. Then we can have the fate of glucose yedu tichitarisa in form of a diagram. Teta kuti isusu glucose yedu can be converted into fats and oils here. That's when it gets to be used up for seeds, right? Then the other one, we are saying that the glucose can be combined with, so it can be combined with nitrates from the soil, and then it can give this amino acid. So this is the structure for amino acid, where we have this one, I think we talked about it when we talked about molecules, the molecules that are found in biology, which are biological molecules. We also talk about amino acids and their structure. And then the glucose can also be converted into sucrose for transport around the plant. So if it is going to go around the plant, it has to go in form of what? In form of sucrose, such that it can be transported around the plant. And also the other glucose is going to be going into the other cells for respiration, for release of energy. So it goes particularly to the mitochondrion. So the mitochondrion are the ones that's the site for respiration. And it so respiration yedu yonoitika muma mitochondrion iglucose inenge ichitase ichibenwa up. Then the other thing, glucose can be made into many molecules joined together. For example, we can have cellulose, and we can also have starch for storage. So why starch? Because starch, it is insoluble, it won't be affected by osmosis in cells. So why? Because since it is insoluble ndizvo haimixi nemvura, because mvura paipe painoita osmosis ndizvo inokwanisa kufamba nezvinhu zvinenge zvakadissolva mairi. So starch yedu is a good way of storing what? Storing the glucose. And also cell walls edu anoshandiswa what? Cellulose. So those are the fate of what? Of glucose, right? Then we have to look at an investigation of photosynthesis. So we are investigating starch production and the need for chlorophyll. And so in other words, we are saying kuti if we know kuti photosynthesis produces starch, because we are saying kuti if photosynthesis produces glucose pekutanga, then we said here that glucose is converted to what? To starch for storage, which means kuti each and every leaf raukuona mukati maro rinenge rakachengetedza what? Starch, right? So we can investigate that starch, and so that starch iyoyo yatinowana, it can be a proof that photosynthesis irikuita sei? Irikuitika. And then we can also after knowing that investigation for starch, we can also see the need for chlorophyll. Is that is this chlorophyll needed for photosynthesis to what? To take place, or photosynthesis is just okay on its own? Is that okay? So on our first point, although plants make glucose in photosynthesis, leaves can not be tested for its what? Presence. So we can't test for the presence of glucose within the what? The leaves. So as glucose is quickly used and converted into other substances and transported or stored as starch. So if we want to see that particular photosynthesis, we're not going to test for glucose, but rather we're going to test for what it stores. It stores starch. So starch is stored in chloroplasts where photosynthesis occurs. So testing the leaf for starch is a reliable indicator of which parts of the leaf are photosynthesizing. So we're saying that chloroplasts, that's the site for photosynthesis, right? That's where photosynthesis occurs. And starch can be stored in what? In these chloroplasts. So since it can be stored in these chloroplasts, we can test those chloroplasts could produce starch here. So starch, that's why it is a reliable indicator. Maybe the question comes here, why is testing for starch a reliable indicator for testing for photosynthesis, right? Or for investigating photosynthesis? So you'd say it is a reliable indicator. Why? Because starch is stored in the chloroplast, which is the site of what? Of photosynthesis. And starch is not soluble. So rather, to test for glucose, we can't test because the glucose is immediately changed to starch. So starch test becomes a more reliable test. Let's go to the next page. Leaves can be tested for starch using the following procedure. So the first thing is that the leaf, so that's number one, the leaf is dropped in boiling water. And the reason why we drop it in boiling water is so that we can kill and break down the cell walls. Are we together? So that we can kill and break down the cellulose. And it, so you find in other textbooks it talks about boiling the leaf in water first of all, so that we can denature enzymes. So when you denature enzymes, we are making that leaf to be in a state whereby rechaite many other what reactions, because enzymes are responsible for chemical reactions, right? So the leaf is left for, on process number 2, the leaf is left for 5 to 10 minutes in hot ethanol in boiling in a boiling tube. So it can be left in what? Boiling ethanol, and sometimes it can be methylated spirit can also be used. So this removes the chlorophyll so that my color changes of iodine can be seen more clearly, because the color change for iodine normally it's from brown to blue black. So if we have that green color, right, we are unable to really see the change, yeah, blue black or brown, right, because the green is a little bit more more denser color. So we have to remove that green. That green, we remove it by boiling the leaf for 5 to 10 minutes in hot ethanol in a boiling tube. And normally we use what we call a water bath, because ethanol is highly flammable. Anyways, we'll talk about it when we look at the next stage, but rather tichingota some stages as they are, and we'll look upon them in detail. So the third stage is that the leaf is dipped in boiling water so that we can soften it. So after we have boiled it in ethanol, then we have to boil it in what? In water, so that we can soften it, such that kuti pateta kuisa zvinhu zvedu zvatino apply ipapo rinenge risina kuomarara, rinenge rava soft, saka we can apply easily. Then the fourth one is that the leaf is spread out on a white tile and covered with iodine solution. So why on a white tile? Because a white tile can clearly give us my visible changes in my colors. So, and then it is covered with what? Iodine solution, which is brown. Then this iodine solution would turn blue black if starch is present. So in green leaf, the entire leaf would turn blue black as photosynthesis is occurring in all areas of the leaf. This method can also be used to test whether chlorophyll is needed for photosynthesis by using a variegated leaf, which is a variegated leaf. We are saying that it is a leaf in two colors, where the other color is white and the other color is green. So the green color no pani chlorophyll, and then pani white pani absence of chlorophyll. So the white areas of the leaf contains no chlorophyll, and then when the leaf is tested, only the areas that contain chlorophyll would stain blue black. Then the areas that had no chlorophyll remain orange brown, as no photosynthesis is occurring here, and so no starch is stored. So this test for starch, as said earlier, is tikatesta for starch yedu ndiro, that test iyoyo inokwanisa kutibetsera futi kuti tikambobvisa chlorophyll chii chinoitika. Is that okay? Tikambochinja sunlight intensity, chii chinoita sei? Chii chinoitika. Are we together? So let's go to the procedure in detail. And so we have the pictorial procedure. So the first stage redu is that we are boiling the leaf in what? In water. And as we are boiling the leaf in water, tishansa Bunsen burner redu, we are boiling this leaf in water such that we achieve an objective to what? To kill the leaf. And the reason why we are killing the leaf is so that we what? We denature enzymes. Is that okay? Saka tikadenature enzymes, no other reactions would occur. And also the reason why we are boiling the leaf or denaturing the enzymes as well is so that we can break down the cell walls. Is that okay? So that's the reason why the first stage has to take place. And the second one, now we are saying on the second one we are then boiling, but now you see that we have this boiling tube. Rather, we are not boiling in alcohol or ethanol directly. Now there's a reason why we don't boil directly. Are we together? We are boiling not directly because we are saying that this ethanol is inflammable. Is that okay? This ethanol can reach 100 degrees, right? With direct heat inovira, getting to flames. But then it can reach using this setup, which is called a water bath. It can reach using a water bath. Would see kuti zvinoricha mbichana but zvichita sei zvichitoita zvakanaka. So the reason why we'll put that water bath is because ethanol is flammable. But the reason for boiling the leaf in alcohol or ethanol is so that we can remove chlorophyll. So to remove our chlorophyll, our ethanol would turn green. Why? Because ethanol here yata say quiver out chlorophyll from the green leaf. Then you quiver out chlorophyll from the green leaf. This green leaf redu rakusara brittle, right? Saka tiri kusara brittle, tiri kusara kuti rakaomarara. Are we together? Then toenda next. The third thing is that the leaf is then washed in water, right? Saka washed in water here, we're just necessarily saying that the leaf is then boiled in what? In water. But this leaf boiling in this, it's for the second time. Are we together? The first time we were boiling the leaf so that we could kill it, or so that we could remove the cell walls. But the second time now we are boiling, and we are boiling such that we make sure that the leaf redu rinobva rava soft. Are we together? Because rinenge raita harden parabviswa chlorophyll, because alcohol inoomesa zvinhu, right? But as you can see now, leaf redu iri rabva from a color which was green, and now the leaf redu ra transparent, ra white. And it so kana ra white kudaro, tikazonzi taedda something on it, we are able to see the color changes zvakanaka kuti ndepapi pari kuchinja what color, right? So on the fourth and fifth stage, we are saying we add iodine solution to that leaf. And so to add the iodine solution to that leaf, the reason is that iodine will change color in the presence of starch. So if it changes color in the presence of starch, it is able to give us detail kuti paitika photosynthesis here kana kuti hapana kuitika, because if it changes in the presence of starch, if it changes to blue black, it means kuti pane paitika chi photosynthesis. Is that okay? Then we go to the fifth stage where we have to observe the color change. And so when we are observing the color change, it turns blue black. If photosynthesis was available, it would turn blue black. If starch was available. Starch kana iri available, it means photosynthesis yakaitika. Then if no photosynthesis kana photosynthesis isina kuitika, it would remain what? It would remain brown. And it saka pama color changes edu tine a negative what? Not negative test. Negative test ndo ire yatiri kuti kune no what? Starch. And saka kana kune no starch, kune no color change. so, or it remains brown. Are we together? Then yatinoti positive test result tiri kuti panenge pane chi starch present, then color change yedu, color change from brown to blue, black. Are we together? So ndo yatiri kuti positive what test and a negative test. So we got lastly to our notes here. Care must be taken when carrying out this experiment. So this is our precaution, right? So precaution. Care must be taken when carrying out this practical, as ethanol is extremely flammable. So at that stage of the experiment, the Bunsen burner should be turned off. The safest way to heat ethanol is an electric water bath rather than using a beaker over a Bunsen burner with an open flame. So we don't have to use an open flame. So what we would use is what is known as a water bath. Then let's proceed. We now need to know kuti is light needed in photosynthesis. We're still doing our investigations. So remember, after any investigation, we'll have to test the leaf for starch. So the test for starch remains our classical test. It remains our classical thing kuti tikazobvisa zvimwe zvatobvisa, we go back to the test for starch. Saka the test for starch, make sure kuti wanyatsoibatisisa. Kana unga usina, replay the video and then wanyatsoibatisa, because the test for starch is what we're going to continuously do. And it so on the need to test for the light, kuti light iri necessary here, kana kuti chlorine iri necessary here, kana kuti any other factor iri necessary. The first thing that we need to do is we need to remove the starch that's already in the plant. And so to remove that starch that's already in the plant, we remove it by putting the plant into darkness for about what 48 hours or 24 to 48 hours. So from the 24 to 48 hours to expect kuti plant redu rinenge raita se rine radya starch yese yarishandisa ndizvozvo rasara muma leaves masisina starch. So if we then remove a factor from there, ndizvozvo takuchikwanisa kuona kuti from there ta remove a factor iyoyo pane here starch ichaita se produce. And so we go to our first point. The same procedure in investigation above can be used to investigate the light if light is needed for photosynthesis. So before starting the experiment, the plant needs to be de-starched, as I was saying earlier on, by placing in the dark cupboards for 24 hours. So sometimes it's 24, and we can just say 24 to 48 hours. So this ensures that any starch already present in the leaf will be used up and will not affect the results of the experiment. Is that okay? So in my question, the plant was not de-starched. So state one error that can be there. It means one error kuti our results will be affected by the starch that's already present. We won't be able to know which one was produced and which one was not. And then the following de-starching, a leaf of a plant can be partially covered with an aluminium foil. So we have this guy called an aluminium foil. This is just a piece of an aluminium, right? So we are saying that we have our leaf here, right? We have this leaf, and then we're going to place an aluminium foil, which is just a covering, a foil that covers. So that foil makes sure that no light reaches to this place inenge yakakavha apa. Tonoona light rinoita sei rinosvika ipapo because aluminium foil doesn't allow light to pass through, right? Then the leaf can be removed and tested for starch using iodine. Saka tikuti tarisa day rese leaf redu but rine aluminium foil, and then tozoribvisa totesta for what? For starch. Then the area of the leaf that was covered with aluminium foil will remain orange brown, as it did not receive sunlight and could not photosynthesize, while the areas exposed to sunlight will turn blue black. They will turn blue black. Why? Because they were exposed, right? They were given enough things to work with, and it then photosynthesis happened. Saka tikuti pese pasina kusvika what sunlight, pese pacharemaina pari pe brown, and then pese pakareceive sunlight pachatena blue black. So this proves that light is necessary for photosynthesis and the production of starch. So let's see again the need for carbon dioxide in photosynthesis. So the first thing that we do is that we destarch the plant, as I have said earlier on, so that we can remove all the starch that is within the plant leaves. Then you tie a clear bag containing sodium hydroxide. So we have this guy, sodium hydroxide. So his purpose is to absorb carbon dioxide. So when it absorbs carbon dioxide, it makes sure that plant redu harina carbon dioxide yarota sei yarikwanisa kuwana. But andapa patashandisa a clear bag, right? A clear bag redu or plastic bag redu riclear richaita kuti light riite through ndirozve, but air iri mukati umomo ichatorerwa oxygen, ichatorerwa inonzi carbon dioxide yayo nenyaya yekuda kwesodium hydroxide yatetiita sei techishandisa. So tie a clear bag containing water. Water yedu iyi iri kushanda as a control, because we are saying that water yedu haikwanisi kuabsorba carbon dioxide. So in other words, mune water mune bag rinenge clear bag rinenge water is the control experiment, is the one that has all factors available. And the reason why we need such a thing called a control treatise is so that we can be able to compare. After we would compare the results, because the control receives every factor and the experiment doesn't receive one factor, and that we are investigating. So abatanzi tie a clear bag containing water, which is the control experiment, which will not absorb carbon dioxide from the surrounding air around another leaf. So we have two leaves here, one rine sodium hydroxide, then two rine water. One redu ndo experiment irozve rine nochi carbon dioxide, then two redu is what we call the control, ndirozve rine all factors that are required. So in other words, mutu medum muchaitika photosynthesis umu mamitike photosynthesis. Reason sodium hydroxide yedu absorb what carbon dioxide. So zvazonzi place the plant, we're on this point, place the plant in bright light for several hours, then test both leaves for starch using iodine test. So if you're using iodine test, the leaf with the bag containing sodium hydroxide will remain orange brown. Why? Because there's no photosynthesis that it's taking place. Then the leaf from the bag, the control bag containing water, should turn blue black, as it had all the necessary requirements for photosynthesis. So that's how we see the need for carbon dioxide for photosynthesis. That's how we investigate it. And then we go to the rate of photosynthesis using a water plant. So here now we are no longer talking about the aspect yekuti ichi chiri necessary for photosynthesis ere ichi chiri chi. Now we are talking about how fast photosynthesis is happening, because this word rate, it means speed. So in other words, we're going to investigate the speed of photosynthesis using a water plant. So a water plant redu, it's a plant that lives in what? In water. Saka plants usually used are a Elodea plant, as well as what? A Camboba plant. They are types of what? Pond weeds. Weeds that grow within the pond. They grow in water, right? They don't require what? Much of the soil. So as photosynthesis occurs, oxygen gas is produced, or is released. And so if oxygen is released as a gas, right? So in other words, we will observe that there will be bubbles of gases as photosynthesis is what is occurring. And so as the plant is in water, the oxygen released can be seen as bubbles leaving the cut end of the pond weed. So the number of bubbles produced over a minute can be counted, and we can record the rate. Saka pakurekoda rate tikutaura kuti mangani ari kuitika per minute. If the rate is slow, we are saying 2 bubbles per minute. If the rate is fast, we can say 100 bubbles per minute. Ndo zvatikureva rate iyo, that's how it can be what measured. We just have to read the number of bubbles anenge aitika per minute iroro, and then would say the number of bubbles divided by the by the time taken, which is the minute, and then we are able to see the rate. So a more accurate version of this experiment is to collect oxygen released in a test tube inverted over the top of the pond weed over a longer period of time, and then measure the volume of the oxygen that is collected. So this practical can be used in the following ways. So before we look at those ways, we are saying that pekutanga tatichiti unoverenga number of gas bubbles, and it can, but kuverenga number of gas bubbles, it's you doing the reading, and it kokapotsa kokwati paunobwera pependo pari kubuda, then wavhura maziso watadza kuverenga what that gas bubble iroro, and it can. So there is room for what? For error, because it's you who's doing the observation uri wega. And then we can make another observation which is more accurate, whereby tinokwanisa kucollecta the gas that has been produced, and then tacollecta the gas that has been produced, then we then measure its volume. Saka tika measure its volume, would make use of the volume produced per a certain time, would still calculate the rate, but in terms of volume, not in terms of the number of gas bubbles. So this practical can be what? Used in what? The following ways. Ndoma ways atachula tichitarisa. Pekutanga, investigating the effect of changing light intensity by moving the lamp. So it's by moving the lamp different distances away from the beaker containing the pond weed. So we want to move this lamp on different distances away from the pond. This is the pond which is holding the what? The weed. So what we are saying is to move what? By to move the lamp a distance. But the reason why we are moving the lamp a distance is we are changing what? The light intensity. So in other words, if the lamp is closer, it has more light intensity. If it is far away, it has lesser light intensity. And so we have this set up. Tingoitarisa just a little bit. Chiona tese. Trukuti susu pekutanga. So this is what you do sometimes, even in exams, and you have to study your diagram. And I say just don't rush to questions, right? Don't rush to questions. Do bichana uchitarisa each and every what part of the diagram, but you are relating it to the information that you haven't. So pekutanga, we have our lamp here. This one, our lamp that's here. Then page 2, we have this glass tank filled with water. And So this glass tank that is filled with water, ndopataisirwa this thing here, and then they said a factor being investigated is what? Is the distance of the lamp that can be changed. Are we together? The distance of the lamp that can be changed. Then that distance of the lamp that can be changed, viands IV, and it. So trukutichi is also in general we know that water yedu allow kuti light rite pass through, and it can. So possibly maybe I'm not so sure, maybe light redu rufana kuenda via the what, via the glass tank, so that it can be concentrated on one area, or turikutaura kuti water yedu iyi since light redu rine heat ndiro water yedu iri kuita sei iri kuita ichitorawo imwe heat to absorb the heat, such that heat haisvike uko kwakanyanyota sei akawandisa. But rather let's see kumberi tabva tanzi pane ka thermometer. Saka ka thermometer kandeke kumonitor what temperature, and it is to monitor what temperature. And why we are trying to monitor the temperature ndizvo is if we change temperature whilst we are changing also intensity, we are unable to see kuti effector ipi yanyatso affector. Are we together? So our thermometer here is to monitor temperature yedu kuti kana iri pa 20 ndizvo kana takuona kuti pane zvakuitika ndizvo the temperature yedu inofanira kuita sei kudzikiswa so that kuti inongoramba iri at a certain what range, and it kana yaita at a certain range ndizvo it doesn't affect the temperature of this, and it because rather tikato affect temperature takuto affect rate ye enzymic activity inenge yakuitika mukati. So we have to what, we have to really monitor that what, that temperature, and it. And number 2, we have an inverted boiling tube. Boiling tube yedu inverted it's so that we can collect the gas here, which is the oxygen. Saka inoita ma bubbles, then it is collected there because it's less dense than water. Then oxygen gas bubbles produced as photosynthesis occurs, CV. Then water with the sodium hydrogen carbonate. Saka water yedu, it contains what? Sodium hydrogen carbonate. Is that okay? So sodium hydrogen carbonate, supposedly it's there such that it can provide this plant with carbon dioxide. Then we also have, because then we can also have photosynthesizing aquatic plant. Yeah, that's the photosynthesizing aquatic plant. So this plant is getting carbon dioxide that is dissolved, and then it uses that carbon dioxide, it produces what? Oxygen. Then the oxygen is collected here. But the main factor that we are investigating here is the light intensity. So the more we draw the light closer to the setup, the more the photosynthesis occurs, and the more we draw the light away from the plant, the lesser the photosynthesis. So we can look investigating the effect of changing temperature by changing the investigating the effect of changing temperature by changing temperature of the water in the beaker. So we are saying that our lamp here now, it is no longer moving, but it is just a source of light. So as you can see from the previous one yedu tachita kuti lamp redu ndori kumova, but we had this thing here. Now I think we now know the purpose is to take away the heat from the what, from the lamp, such that this lamp heat yayo haise heat yayo kune setup head, and it. So at this process now reached 2, we are saying that the source of light is just constant, it is at one place. But isusu takuzvisa pane a hot plate ndizvo, such that we can change temperature. Saka kuya ku previous tachimonitor temperature so that temperature remains constant, and then this time we want to change what, the temperature. We want to change the temperature. Saka hot plate yedu ichachinja what, temperature. Saka painochinja temperature, it will affect enzymic activity, and it. then affecta enzymic activity tichaona kuti photosynthesis yedu inokwanisa kumbotanga ichiita zvakanaka. Then pane optimum temperature inoreacha, which is the highest, where we can have the Vmax, which is the maximum of the photosynthesis that's occurring. Then from there, from the optimum ikaramba ichiwedzerwa now takutotanga kutouraya plant redu, and the enzymes will begin to deteriorate, or they will begin to be denatured. Then hatichakwanisi kuwana the same results as we were getting previously, and then we can also look at the other one. We are saying we are investigating the effect of changing temperature on the rate of photosynthesis. So okay, let me just see. Okay, so investigating the effect of changing carbon dioxide concentration, and so we are not looking at this one. Yeah, investigating the effect of changing carbon dioxide concentration by dissolving different amounts of sodium hydrogen carbonate. So as I've highlighted earlier on, we're saying sodium hydrogen carbonate is a source of what? Carbon dioxide, but this is a source of dissolved carbon dioxide, so we can say that it's aqueous carbon dioxide that's dissolved in water. And so this time we are trying to make sure that we have a glass tank filled with water. This one would keep our temperature constant, and then this one it would keep our light intensity what? Constant, but now chinja here is now we are changing the amount of what? The amount of sodium hydrogen carbonate. So as the amount of sodium hydrogen carbonate increases, we'll find out that the temperature, the rate of gas bubbles that are produced would also increase up to a particular level. So we are saying that when all the leaves and all the cells are photosynthesizing, right, would have reached the maximum what? Velocity, would have reached the maximum speed at which photosynthesis can occur. So nyango ukazowedzera the amount of carbon dioxide, nyango ukazoiwedzera it might have no effect, because all of the cells anenge achita what? Achita photosynthesize, and you can't go beyond. Then investigating the effect of changing carbon dioxide concentration on the rate of photosynthesis. Care must be taken when investigating a condition to keep all other variables constant. Saka to keep all the other variables constant, what does it mean? Tikuroti kana tichitesta for carbon dioxide, which means kuti tichati temperature inofanira kunge iramba iri the same, light intensity inofanira kuramba iri the same, haifani kuchinja. This then ensures that we have a what? A fair test. For example, when investigating the changing light intensity, a glass tank should be placed between the lamp and the beaker to absorb the heat from the lamp, so that we avoid changing the temperature of the water. So glass tank redu rakaiswa rere, issue yaro yanga iri kuita sei? To absorb what? The heat, such that patiri kuchinja what? Inonzi patiri kuendesa inonzi the negirikini redu, the lamp redu towards the what? The plant, closer to the plant, harisi kuzochinja temperature yechi inenge irimo musetup yedu yechi yeka conduit kedu. So that glass rino absorber temperatures. Then after having known all these, now we want to now understand kuti kana tine ma-factors aya, and these factors are operating ndizvo, there's a limit to which things can have ndizvo. So we can't necessarily say that the increase of light intensity inongoramba ichingowedzera rate ndizvo. It will go to a certain limit. The limit, the increasing temperature, it will go to a certain what? Limit ndizvo. So we want to understand limits that are there, and also what factors will be limiting factors, because there are other factors asinachi a limit. Tichitaura kuti haana chekuita nekuchinja speed yechi photosynthesis in its occurrence. And So if a plant is given unlimited sunlight, carbon dioxide, and water, and is at a warm temperature, the limit on the rate or speed at which it can photosynthesize is its own ability to absorb these materials and make them react. Are we together? So yapiwa zvinhu izvi in unlimited form. Handitika saka painenge yapiwa zvinhu in unlimited form zvese zviripo haizvipedze, what becomes a limiting factor is its ability to absorb those things. So if it can't have a speed yakanyanyisa yekuabsorba, that speed yainayo is the one that will limit what? The reaction. And it, however, most plants do not have unlimited supplies of their raw materials, so their rate of photosynthesis is limited by whatever factor is the lowest at that time. So what is limiting? What is limiting? I want us to go to this point. Whatever factor is lowest at that time. Whatever factor is lowest at that time becomes the limiting factor. So a limiting factor can be defined as something present in the environment in such short supply that restricts life processes. So now this can be a little bit complex, and it can yavatipa as a definition, and oh, that's okay. But a limiting factor, it's whatever factor that is lowest at that time is the one that we call a limiting factor. So there are three main factors which can limit the rate of photosynthesis, which is temperature, light, as well as carbon dioxide concentration. So we are saying carbon dioxide concentration to tell carbon dioxide yakawanda say, and so although water is necessary for photosynthesis, is not considered as a limiting factor. Why? Because it is abundant. There is no plant that can live without water. Saka kuti photosynthesis itonzonzi yatozoitika, it means the water already is in its available what state. Dai yanga isipo, then the plant ringadai rakatofa risina kwanisa kuzoita photosynthesis. So we don't necessarily mention water as a limiting factor. Are we together? So we go to temperature. So as temperature increases, the rate of photosynthesis increases as the reaction is controlled by enzymes. So the reaction here, it is controlled by my enzymes. So since it is controlled by enzymes, we are saying that how enzymes behave with temperature is what will then happen. And so however the reaction is controlled by the enzyme, this trend can only continue up to a certain temperature, and beyond which the enzymes can begin to denature and the rate of reaction decreases. Is that okay? So sanke zvatakaita when we looked at enzymes, right? We are saying temperature, right? So let's say if the enzymes, they have an optimum temperature of, let's say, around 30 degrees, right? So you're saying that's where they have the highest what rate. So we're saying rate is here. So if they have the highest rate there, then we have the rates decreasing, and the rates, they are decreasing on the other side, right? So we're saying as we increase the temperature from 0 up to 30, we see that the rate of photosynthesis will be increasing, but up to a certain level, which we call the optimum temperature, right? So after the optimum temperature, when we continue increasing the temperature here, the rate begins to decline. Why? Because enzymes begin to denature. Saka paanta begin to denature, it means my enzyme akuta mashoma, saka rate akuta ishoma. Kunoku ma enzymes atova mashoma because akawanda achona they have denatured. Kunoku kuzosvika pa point yekutoti hapasina enzyme raakuita sei raakufunctiona kuti tese speed up the rate of reaction, and then we see that our rate of reaction would drop significantly. Then we go to look at possibly the graph itself, right? So that's the graph rakadafananira tatiita. As the temperature is increasing, right, the number of substances that are colliding between each other are kuwedzera, and the enzymes increase. So the number of collisions anokonzeresa kuti pave ne-effective what collision. Then effective collision yedu inokonzeresa kuti padzikiswe iyo inonzi chi activation what energy. Ndo zvinoita maenzymes. Adzikisa activation energy, rate yedu inoita sei? Inowedzera, ndizvo, but inowedzera up to an optimum temperature. After an optimum temperature, they will begin to denature, and then it would have a significant what? Drop. Then we look at light intensity. So the more light intensity the plant receives, the faster the rate of photosynthesis. So this trend will continue until another factor required for photosynthesis prevents the rate from increasing further, because it is now in short supply. So atikuti light intensity yedu icharamba ichiwedzera iyo ine sharp curve. So rather it was supposed to continue going like this, because the more the light, the more the energy, the faster it has to be, ndizvo. But tikutaura kuti panapa pari kusvika at this point, ndizvo. Another factor riripo rakubva raita come into play, ndo rakubva raita kuti rilimite manje, and then our graph becomes what? Straight like this, ndizvo. So let's say kuti another factor maybe temperature ndo inobva yatanga kulimita, kana kuti the other ones that what begin to limit, and then graph redu robva raita straight. So unlike remaenzymes rikuti rikubva radzika because ari kuita denature, rino rinobva raita straight, robva rangoita a constant what? Speed. That constant speed iri kukonzereswa nekuti tava what? Another factor which is coming into play, and then that other factor, it's becoming the limiting what? Factor. So continuously increasing would not continuously increase the graph. So at the end of the day, we are having a graph redu richisara rakaita se richingosara rakangodai. Are we together? Then twenda pane the next one, effect of light intensity on the rate of photosynthesis. So light intensity yedu at low light intensity, increasing the intensity will initially increase the rate of photosynthesis. So at a certain point, increasing the light intensity stops the increasing the rate. The rate becomes constant regardless of how much light is being increased, as something else is the limiting factor. So that's what we're talking about. So the factors which could be limiting the rate when the line of the graph is horizontal or parameter constant include temperature not being high enough or not enough carbon dioxide. So temperature and carbon dioxide concentration, they come into play, and then tova tawana tane just a constant what? Rate. Then carbon dioxide concentration is another factor that we investigate on the rate of photosynthesis. We're saying carbon dioxide is one of the raw materials required for photosynthesis, for photosynthesis to take place. Then this means that more carbon dioxide, this means the more the carbon dioxide that is present, the faster the reaction can occur. This trend, however, can continue until some other factor required for photosynthesis prevents the rate from increasing further, because it is now in short supply. Saka the graph rate we expect it to be the same as the graph rate light intensity, and it so it will be the same. Saka rinongo kwidza up to a certain point, it becomes constant. Then the effect of carbon dioxide concentration on the rate of photosynthesis. The factors which could be limiting the rate when the line of the graph is horizontal or is constant include temperature not being high enough, as well as light being not enough. Then we go to the changing greenhouse conditions. Changing greenhouse conditions. So the knowledge about limiting factors and how they affect the rate of photosynthesis can be used to help control factors in the greenhouses to ensure maximum crop yields for farmers. And so they can see the limiting factors. That knowledge for limiting factors, it can help them to maximize crop yields. So growing crops outside does not allow the farmer to control any of these factors to increase the growth for plants. Is that okay? So in the greenhouse, several conditions can be manipulated to increase the rate of photosynthesis, including number one, we can have artificial heating. So artificial heating, enzymes controlling the photosynthesis can work faster at slightly higher temperatures, only used in the temperature countries such as the UK. Then the artificial, number two, we can have artificial lighting. Plants can photosynthesize for longer. So they can be artificial light, right? So most people, they just think kuti light rinoita kuti photosynthesis iitike light rezuva. Just think that it is only zuva, but no, we can have artificial light. And that artificial light rinoita kuti yanonzi maplants edu, they can photosynthesize for a longer time. Mamwe akuenda mudarkness, but mamwe arikungoramba ane light. So achiramba ane light kudaro, they are keeping on photosynthesizing. And then number three, increasing carbon dioxide content of the air inside the plant can also help these plants photosynthesis quicker. Saka kana kuri kunze kuatmosphere tikawedzera carbon dioxide, it easily gets lost, right? But in greenhouses, cover, we are controlling the what? The environment that's inside. We're controlling the carbon dioxide concentration. So if we increase it, we can have an increase. And so as you can see, pai patamboita an experiment whereby we were testing why whether carbon dioxide is necessary. And taisa inonzishi, we placed sodium hydroxide such that it absorbs what? Carbon dioxide pakaswa plastic. And it pa greenhouse chine chakaita kunge chiplastic futo chakakava that house. So in that house chine chakakava umomo, we can put certain things, that certain chemicals that can then release carbon dioxide into the air that is around that place. So as we do that, we increase the carbon dioxide concentration. Then the fourth one, regular watering. So the plants that are living more natural environment, they require watering here and there, and some of them, they would just use rainwater, and manganishaniswa irrigation. But in greenhouses, we are not saying that we are waiting for water to rain. Water is what is then put into those greenhouses via irrigation, and they only receive irrigation water, not receive what? The water from the sky, panitka rain. Then when considering the use of greenhouse and manipulating conditions like this, farmers need to balance extra cost for providing the heating, lighting, and carbon dioxide against the increased income. So at most times we find out that these greenhouse gases, they are mainly used for those people that are farming zvinhu zvakaita semaruva, because maruva, tichitaura kuti maruva edu ane a higher margin yaunokwanisa kuwana rather than what? Chibage. Let's say waita chibage chako wochiisa mugreenhouse, greenhouse iyoyo dai zvainzi yangaita maruva yaikupa mari yakawandisa kudarika chibage. Then in tropical countries where temperatures are much hotter, greenhouses may still be used to control other conditions. However, they may need to be ventilated to release hot air and avoid temperatures rising too high, which could cause the denaturation of enzymes. So controlling photosynthesis reaction will be required. And then takwenda pane the effect of light on net gas exchange. Effect of light on net gas exchange. So when you talk about net gas exchange here, we're now talking about these guys which are called what? The guard cells. And we're saying here we have two guard cells, and pakata papa we have an opening called the what? Stoma or stomata. And so we have guard cells. So in other words, gaseous exchange yedu iri kuitika ipapa apa tiri kubudisa magases tichiisa magases inside, and but it's via the stoma. But these guard cells, they can be affected by light intensity. That's what we want to discuss, right? And so plants are respiring at all times. So plant cells are taking in oxygen and releasing carbon dioxide as a result of aerobic respiration. So plant also photosynthesize during the during daylight hours, for which they need to take in carbon dioxide and release oxygen that is made. Then at night, the plants do not photosynthesize, but they continue to respire, meaning they take in oxygen and give out carbon dioxide. So that's what happens. We are saying this is during the day where photosynthesis is taking place. Then during the night we have respiration taking place. So we have photosynthesis during the day and respiration during the night. So during the day, especially when light is bright, plants are photosynthesizing at a faster rate than they are respiring. So the net intake of carbon dioxide there's a net intake of carbon dioxide and a net output of oxygen. So we can investigate the effect of light on net gas exchange in aquatic plants using the pH indicator such as sodium hydrogen carbonate indicator. So this is possible because carbon dioxide is acidic gas when dissolved in water. So sodium hydrogen carbonate indicator shows the carbon dioxide concentration in the solution. So the table shows the color that the hydrogen carbonate indicator turns at different levels of carbon dioxide concentration. So this experiment particularly toda kuona kuti inonzi kuti net exchange yedu yakamira sei, and saka in other words we are trying to see the concentration of carbon dioxide kuti iri kuchinja sei as we change from daytime to what? To night time. So when it has the highest concentration, it's yellow. Then when it has the lowest concentration, it's what? It's purple. But it turns from yellow, red, then yellow, orange, then red, then magenta to what? To purple. Those are the color changes. So when it is orange and yellow, there is more respiration. More respiration is greater than photosynthesis, so there is a lower pH. Then the atmospheric level where photosynthesis is equal to respiration, that's where we get it as red. And then at where we have more photosynthesis taking place and photosynthesis is lesser than respiration, we have a more alkaline. Why? Because we have less CO2. Here we had more CO2, and we had said earlier on CO2 is an acidic gas, so it's a more acidic solution. So that's why we have the highest indication of carbon dioxide concentration. So that's why we have yellow and red. And there we have the lower because there is less oxygen, less carbon dioxide, because there is more photosynthesis taking place, right? So to put photosynthesis can take place more, carbon dioxide here is being used up, right? So can used up, we are saying that the carbon dioxide concentration inodzikira. So let's say, for example, you want to decrease the carbon dioxide concentration within an area, you plant green plants. That's why the green plants are even planted as a result yekuti techida kuda kuavoida greenhouse what gases. Panonzi plantai trees, plantai trees. Why? Because trees would take out that what carbon dioxide, and it then tender next. Several leaves from the plant are placed in stopped boiling tubes containing some hydrogen carbonate indicator. Then the effect of light can be investigated over a period of hours. So the results from the typical gas exchange experiment are shown. So we have test tube A, B, and C. We have the contents. A, it is the leaf, and the conditions that we are testing is light. So if there is light, it means that there is photosynthesis that's going to take place. Here we have photosynthesis taking place. So in other words, a lot of carbon dioxide has been taken in, and the carbon dioxide concentration would decrease. That's why the indicator then turns purple to show that there is a low concentration of carbon dioxide. Then in conclusion, would say that there is a net intake of oxygen by the leaf. There is a net intake of oxygen. There's a net intake of oxygen by the leaf in the light. This one. Let's look at the second one, but this one is still having some questions there. Then biachona tovatans isusu, there is a leaf, right? And then the leaf is placed in the dark. So whilst it's placed in the dark, we are saying that there is respiration. So respiration is occurring, and once respiration occurs, tukutaura kuti pane more CO2 and it produced, and it then paitika more CO2 produced, right? It then turns what? Yellow, to show that we have more concentration of what? More concentration of CO2, and then here then they say that there's a net intake of carbon dioxide by the leaf in the dark. Well, I thought personally that maybe they wanted to say that there's a net intake of CO2 here, because CO2 is the one that is being taken for photosynthesis to take place, right? So that's what I thought. Maybe the net intake, maybe they're trying to say what remains more. So what remains more is oxygen. I'm not so sure. Then there is a net intake of carbon dioxide. More concentration of carbon dioxide. There was more intake of what? Of oxygen. So the net, maybe the net we're saying chirikusara. The net maybe we're saying chirikusara. So if chirikuti chirikusara, that becomes correct to say that there is a net intake of what? Of oxygen. So there is a net chirikusara chakawanda. It's what? Oxygen. Panoitika photosynthesis, because though produce what? Chirikusara chakawanda panoitika respiration. That's what? That's carbon dioxide. Then here we have no leaf, and we have light, and we have the indicator turns red. Then this is the control. The two other tubes can be compared with it. So this C was just a control to compare. Then we look at the structure of the leaf. So in the next lesson, since this lesson was quite long, in the next lesson we'll look at plant nutrition, and particularly we'll be starting to look at the structure of the leaf and how it is adapted for photosynthesis to take place. So I'll catch you in the next lesson, and we'll look at the structure of the plant.